Okay, thank you everyone for being here for this session. So apologies for the last minute preparation. I only found out on Friday that I'm giving this introduction talk, but I think it should be all right. So we have two young promising scientists today presenting with us their work on graphene. And I have a humble introduction here to do about graphene. So I come from a background where I worked for carbon nanotubes for many years, and graphene is a promising new area that everyone is talking about. So we really need to find out whether it holds the promise that everyone expects or not. So there are a lot of studies to be done still in this field. So is it carbon nanotubes or graphene? What do we really want? So we are a group on nanomedicine at King's College London, and we work with a range of materials such as protein particles, liposomes, carbon nanostructures, including graphene and nanotubes, and also hybrid materials made either of organic and inorganic material or polymers with inorganic particles. So it fits nicely with the different themes of other particles. And the main aim is to use them as delivery carriers to either small drugs, radioactivity, or nucleic acid. So it's a difficult question to choose from, so which one is the best? And the answer is really, it depends. So you can think of where you want to deliver them, what properties you prefer, and which materials gives you the better properties in vitro and in vivo. So what applications we use them for? So it's mainly drug delivery, and we're interested in three main aims. So one is in cancer delivery. We're also working on using them to deliver drugs to brain cells and across the blood-brain barrier, and also as cancer vaccines, as delivery systems to antigens and adjuvants. So this is where graphene fits. So it comes from this material graphite, and you can appreciate that there are many forms of carbon, ranging from very irregular amorphous carbon to highly organized nanotubes or graphitic structures. So it's all made of carbon, but the size and the properties of the material make them hugely different, and we need to understand what's happening. So graphene has been proposed for several biomedical applications, and one of them is drug delivery. So there are many parameters that we have to consider. The general concept is you really want to break the graphite or the several layer graphene structures into fewer layers that are smaller in size and probably more biocompatible. So we've done quite a few studies over the past few years, and they are all focused on making the material and characterizing them before we are able to employ them for delivery of the drug, because simply failure can happen if we don't understand the property of this material. So here what we wanted to achieve is actually deliver some drug molecules and at the same time break the graphite into smaller water dispersible material. So we used the simple method of combining the two components and using dry milling in a solvent-free manner. And by doing so, we're hoping that we are exfoliating the graphite layers and making more fewer layer mesosheets that can be used for drug delivery. So simply you use a mix and melt, and you don't have any solvents because neither of the materials in this case like the solvents. And we tried different conditions and found the best condition that actually gives you a water dispersible material. So if you look at the milled graphite alone, it does not disperse in water. Even if you mix the graphite alone, mix the drug alone, mill them separately and mix them, they also don't show good dispersion. But actually, if you melt them together, you tend to have a stable dispersion. So looking at how this affects the properties and how much defects you introduce to the material. So you have the characteristic D and G bands, and D bands reflects the number of defects of the material. So you can see upon milling, you're actually increasing the D band compared to the graphite, which is expected, and that tells us you are actually introducing some defects to graphene. But also what it tells us is we are able to form few layers by looking at the 2D band. We're forming few layers graphene compared to the graphite. So we know by doing so, we are able to reduce the size of the graphite into fewer layer graphene, but we're also introducing some defects. So we cannot make graphene, but it's actually graphene with some defects. So far, we don't know much about the size of this material, so we actually call them mesosheets afterwards. And this is a STEM imaging and atomic force microscopy. And you can see that here you have a sheet which is approximately, that's a 1 micrometer bar. But if you have the graphite milled without the drug, you see huge bulks of material that are actually very big, and that's the thickness of the material, compared to much fewer sheets when you have the drug milling the graphite. So these are, again, STEM images. You can see the folding of the material here. You can confirm also the presence of the drug aggregates on the mesosheets. And by looking at the yields spectrum, you can also confirm that you maintain the graphitic structure of graphene layers. We compared this exfoliation method with a standard method called Hammer's method, where it's widely known you introduce huge amount of defects. And by comparing the oxygen to carbon atom percent, you can see that our method introduces much lower defects compared to the Hammer's method. So this kind of the information that we obtained, but unfortunately what we found at the end, despite the formation of good material, we found that the drug here is inactive. So do we use this method to deliver drugs, or do we use the drug to exfoliate graphene? That's actually a question that's open to different applications. Then the second approach, we wanted actually to deliver imaging agents using these water-soluble in this case. So we use water-soluble graphene oxide material that we produce in high scale using modified Hammer method developed in our lab. And again, by using a solvent-free mechanochemistry approach, you melt the water-insoluble spion, in this case, oleic acid coated, with the water-soluble or dispersible graphene oxide. And we use the design of experiment approach to optimize this method to get the sheet or hybrid material of best characteristics. So you can see here, so spion without the graphene oxide wouldn't disperse in water, but actually when you have the hybrid, they form stable suspension that's magnetoresponsive. We can use the thermogravimetric analysis to quantify the content of spion in the hybrid. So these are TM images, show how the spions are associated with the graphene oxide sheets. These are some AFM images. Again, the green color indicates you have more aggregation compared to just flat graphene oxide material. The material was not toxic to a couple of cell lines we tested up to 72 hours, and they showed a dose response MR effect. So obviously there is still more to be studied on the in vivo efficacy and the biocompatibility of the material. And then we wanted to move closer into putting this on cells, injecting this into animals, and actually using them for targeting cancer cells. And at this stage, we didn't even know if we are able to target cancer cells, because we hoped to, but we were not sure. So we have developed some chemical approaches to introduce functionalities to graphene oxide. And I think this is quite important because everyone is talking about the conjugation, introduction of biomolecules, the protein corona effect. So you really have to have controlled functionalities that, you know, you have chemical introduction of the material as opposed to physical adsorption. So what we have here is the graphene oxide with alkyne groups, and by using again the solvent-free mechanochemistry approach, we introduce some linkers with via click chemistry, and we have the melamide group that's amenable for modification depending on what you want to have. So in this case, we have the antibody against MUC1 receptor. So this is the chemical scheme summarizing the production of the graphene oxide, and actually here we can get the material in grams level. So we established a protocol from the Hammers method where we don't have issues with the scale of the material. And that's just a representative image showing the functionalities that you have on graphene oxide, and here we call them nanographene oxide because we reduce the size by sonication. So what you do next is you introduce by esterification the alkyne group. That this is all done in solution based, and then you have the solvent-free method, which has its own advantages obviously, and you have the PEG chain with the terminal melamide. We have a long spacer here because we don't want direct adsorption of the PEG of the IgG on the graphene oxide. And that is important to maintain the activity, the targeting activity of the antibody. So after confirmation of the introduction of these functionalities by FTIR, so here we use indirect conversion of the alkyne into the hydrogen into deuterium, and then we confirm that we have actually also the PEG molecule. It's quite important to confirm the introduction of all these molecules because we find in many cases we end up having non-covalent interactions with these macromolecules. They like to adsorb many molecules. So then you incubate them with cells that are positive for the MUC1, in this case breast cancer cells, and this is just the nuclei. This is the staining of the antibody, and you can see the cells here were incubated just with the antibody alone, but here with the antibody conjugated to the graphene oxide, and you can see there's help of internalization of the antibody into the cells. We image the graphene oxide directly by relying on their photoluminescence properties, and you can see nice co-localization between the green signal of the graphene oxide and also the antibody. So we could confirm actually if you functionalize the graphene oxide, reduce their size, you are able to achieve targeting of the cancer stem cells. So this is all in vitro. What happens in vivo, that's something also to look into. I'll leave this to our next speaker. So we developed then more functionalized material with double clickable group. So that's something May will talk to you about it. And these are really open questions that we ask ourselves all the time in the lab, how to overcome these challenges before we are able to answer and address questions. So we need to have good yield of the material, how to size them in a high throughput method rather than just relying on AFM images, which is quite tedious. Size is important. We have a variably dispersed size, and I think this is very important to control before we draw broad conclusion on the toxicity or have to say that, you know, carbon materials are immunogenic or any of these statements. We need to decide on what size for which application. Am I injecting this into the body? Am I doing this in implants? These are all to be decided. And we need to consider every time we improve the water dispersibility how much of the identity of the material I'm remaining, because modifications may end up having a material that's completely different. It will be nice to have quality control protocol that everyone can stick to standardize this material. I think I'll stop here and I'll probably leave more room for our two speakers of today, and we can ask them questions at the end. Thank you very much.